CSI CSIRNet JRF 2016. Which of the following cell types has the ability to differentiate into all cell types in the human body except for extraembryonic tissues? A. Totipotent. B. Pluripotent. C. Multipotent. D. Unipotent. Answer. B. Pluripotent. CSIRNet JRF 2017. What is the defining characteristic of a multipotent stem cell? A. It can differentiate into any cell type. B. It can differentiate into all types of blood cells. C. It can differentiate into only one type of cell. D. It can form any cell type within the body and the placenta. Answer. B. It can differentiate into all types of blood cells. These questions test understanding of the levels of potency and the differentiation capacity of stem cells, which are essential topics in developmental biology and are highly relevant for CSIRNet life sciences preparation. You said. Commitment. ChatGPT said. Commitment in developmental biology. Commitment in developmental biology refers to the process by which a cell or a group of cells becomes progressively restricted in its developmental potential and starts to differentiate into a specific cell type. This concept is closely related to the idea of cell fate determination, which is a key aspect of development. Commitment involves the internalization of molecular signals that drive cells toward a particular fate and is a critical step in the development of multicellular organisms. Commitment occurs in two stages: specification and differentiation. Specification and differentiation. Biology. Commitment in developmental biology refers to the process by which a cell or a group of cells becomes progressively restricted in its developmental potential and starts to differentiate into a specific cell type. This concept is closely related to the idea of cell fate determination, which is a key aspect of development. Commitment involves the internalization of molecular signals that drive cells toward a particular fate and is a critical step in the development of multicellular organisms. Commitment occurs in two stages: specification and differentiation. Specification and differentiation. Specification is the first stage of commitment. It refers to the ability of a cell to develop into a particular cell type in a neutral environment. At this stage, the cell is said to be specified for a certain fate, but it can still be influenced by external signals, and its developmental path may change if it is placed in a different environment. For example, a cell in the early embryo may be specified to become a muscle cell, but if the cell is moved to an environment where it receives signals for becoming a nerve cell, it could potentially adopt that fate instead. Differentiation represents a more committed state. In this stage, the cell becomes irreversibly committed to a specific fate and begins to express genes that are characteristic of the targeted cell type. Differentiation involves changes in gene expression that lead to the morphological and functional characteristics of the cell. For instance, once a stem cell becomes committed to becoming a muscle cell, it will differentiate into a myocyte, expressing. In this stage, the cell becomes irreversibly committed to a specific fate and begins to express genes that are characteristic of the targeted cell type. Differentiation involves changes in gene expression that lead to the morphological and functional characteristics of the cell. For instance, once a stem cell becomes committed to becoming a muscle cell, it will differentiate into a myocyte, expressing muscle-specific proteins such as actin and myosin, and will ultimately take on the form and function of muscle tissue. Types of commitment. Commitment can be autonomous or conditional, depending on the mechanism that regulates the cell's fate determination. Autonomous commitment. This form of commitment occurs when the cell's fate is determined by intrinsic factors within the cell. These factors could include the asymmetric distribution of cytoplasmic molecules, which influence gene expression. Commitment can be autonomous or conditional, depending on the mechanism that regulates the cell's fate determination. Autonomous commitment. This form of commitment occurs when the cell's fate is determined by intrinsic factors within the cell. These factors could include the asymmetric distribution of cytoplasmic molecules, which influence gene expression and drive the cell toward a specific fate. This kind of commitment is typically seen in early embryonic development when the fate of the cells is largely determined by their position in the embryo and the localized proteins and RNAs that are inherited during cell division. Conditional commitment. In contrast, conditional commitment relies on signals from the surrounding environment or neighboring cells. These external signals can include growth factors, hormones, and cell-cell interactions. Conditional commitment is more flexible and is often seen in later stages of development, where cells in a tissue are influenced by neighboring cells or external cues. For example, cells in a developing limb may be specified to become bone or muscle cells, depending on the signaling environment they are exposed to. Factors influencing commitment. Several factors contribute to the commitment process. Transcription factors, proteins that regulate the expression of specific genes involved in cell differentiation. These transcription factors can be activated by signaling pathways or other cellular cues. Signaling pathways, extracellular signals, such as WNT, Notch, Hedgehog, and TGF-beta, can direct the fate of cells by triggering specific intracellular pathways that activate or suppress the expression of certain genes. Epigenetic modifications, changes in DNA packaging, such as histone modifications and DNA methylation, also play a critical role in determining cellular commitment by influencing gene expression patterns without altering the underlying DNA sequence. Commitment is a crucial aspect of developmental biology because it shapes the proper formation of tissues and organs during development. Misregulation of commitment can lead to developmental disorders or diseases, such as cancer, where cells may become erroneously committed to proliferating uncontrollably. Commitment in stem cells and regenerative medicine. Stem cells are highly relevant in the study of commitment due to their ability to differentiate into multiple cell types. The process of commitment in stem cells is tightly regulated and can be manipulated to direct stem cells to differentiate into desired cell types for therapeutic purposes. This is particularly important in regenerative medicine, where stem cells are used to regenerate damaged tissues and organs. Understanding the mechanisms of commitment in stem cells allows researchers to optimize protocols for directing their differentiation in a controlled and effective manner. In conclusion, commitment is a fundamental concept in developmental biology, representing the process by which cells progressively become restricted in their developmental potential. It involves both intrinsic and extrinsic factors and is crucial for the proper formation and function of multicellular organisms. Studying commitment at both the molecular and cellular levels is essential for understanding development and for applications in stem cell therapy and regenerative medicine. Previous year questions. CSIRNet JRF 2019. Which of the following best describes the process by which a cell becomes progressively restricted in its developmental potential? A. Differentiation. B. Specification. C. Commitment. D. Morphogenesis. Answer. C. Commitment. CSIRNet JRF 2017. During development, the stage when a cell is able to express its characteristics but still retains the potential to switch fates depending on the environment is known as a differentiation. B. Specification. C. Commitment. D. Totipotency. Answer. B. Specification. These questions focus on the stages of commitment and its role in development, specifically highlighting the concepts of specification and differentiation. These topics are essential for understanding how cells are programmed during embryogenesis and development, which is crucial for exams like the CSIRNet JRF in life sciences. You said. Specification. ChatGPT said. Specification in developmental biology. Specification is a critical concept in developmental biology that refers to the stage during embryonic development when a cell or a group of cells becomes determined to follow a particular developmental path, even though it has not yet fully differentiated into its final cell type. Cells that are specified have the potential to develop into a particular cell type or tissue under certain conditions, but their fate is not yet fixed and can be influenced by the surrounding environment. Unlike fully committed cells, specified cells retain a degree of developmental plasticity and can change their fate if exposed to different signals. Specification is often the first step towards differentiation, although the fate of specified cells can still be reversible depending on the cellular context. Mechanisms of specification. Specification involves the regulation of gene expression in response to both intrinsic and extrinsic factors. It is closely related to cell-cell communication, positional information, and the presence of signaling molecules in the embryonic environment. Specification is categorized into two main types: autonomous specification and conditional specification. Autonomous specification. This type of specification occurs when the fate of a cell is determined by intrinsic factors, often localized determinants within the cell. These determinants are usually specific molecules, such as proteins or RNAs, that are unequally distributed within the cytoplasm of a cell during early division. These molecular determinants guide the developmental fate of the cell even when the cell is isolated from its neighboring cells. For instance, in some animals, the zygote undergoes a series of cleavages that result in the unequal distribution of mRNA and proteins, which influences the future developmental fate of the cells. Conditional specification. Unlike autonomous specification, conditional specification depends on interactions with neighboring cells or the external environment. In this type of specification, the cell's fate is influenced by signals from surrounding cells or positional information. An example is the development of the Drosophila embryo, where the segmentation of the body relies on signals from the anterior-posterior axis, and cells are specified to form particular structures based on these gradients. Conditional specification is often observed in vertebrate development, where cells may develop into different tissues depending on the signaling molecules they receive from their neighbors. The classic example of this process is the induction of neural tissue from ectodermal cells by signals from the underlying mesodermal tissue. Role of signaling pathways. Specification is tightly regulated by a variety of signaling pathways that govern cell fate decisions. These signaling pathways include well-known ones such as the WNT, Notch, HETOG, and TGF-beta pathways. These signals often create gradients in the developing embryo, providing positional cues that help cells determine their fate. For instance, in the development of the Drosophila embryo, the bicoid gradient provides positional information that specifies anterior and posterior fates in the developing fly. In vertebrates, signals like sonic hedgehog, SHISH, and bone morphogenic proteins, BMPs, help specify various cell types in the neural tube and limb buds. Specification in stem cells. In the context of stem cell biology, specification is an important concept in understanding how pluripotent stem cells, such as embryonic stem cells, ESCs, acquire specific fates when exposed to certain signaling environments. ESCs are initially pluripotent, capable of giving rise to all cell types of the body. However, when placed in specific culture conditions or exposed to specific signaling molecules, these cells can be specified to become particular cell types, such as neurons, muscle cells, or blood cells. The process of specification in stem cells is essential for both developmental biology and regenerative medicine, where understanding how to control cell fate can be used for therapeutic purposes, such as tissue regeneration and repair. Specification versus differentiation. It is essential to distinguish specification from differentiation. Specification refers to the process by which a cell is instructed to follow a particular fate, but it can still change fate if exposed to the right signals. In contrast, differentiation is the process by which a cell undergoes changes in gene expression that lead to the acquisition of a specific cell type's characteristics, and this process is generally irreversible. While specification marks the initial commitment of a cell to a particular fate, differentiation marks the completion of that process as the cell takes on the specific functional and morphological traits of its designated cell type. In conclusion, specification is a key early event in developmental biology that determines the fate of cells. It can be influenced by both intrinsic factors, like cytoplasmic determinants, and extrinsic factors, like signaling from neighboring cells. Specification leads to the establishment of cellular identity and lays the foundation for further differentiation processes. Understanding specification is critical for developmental biology, stem cell research, and regenerative medicine, as it provides insights into how cells are programmed to form diverse tissues and organs. Previous year questions. CSIR NET JRF 2018. Which of the following is true for specification in development? A. It is a process where cells acquire their final differentiated state. B. Cells become irreversibly committed to a specific fate. C. It is a reversible process that determines the developmental potential of cells. D. It occurs only in stem cells and not in differentiated cells. Answer. C. It is a reversible process that determines the CSIR NET JRF 2017. In which of the following processes does the fate of a cell depend on interactions with surrounding cells? In which of the following processes does the fate of a cell depend on interactions with surrounding cells? A. Autonomous specification. B. Conditional specification. C. B. Conditional specification. C. Differentiation. D. None of the above. Answer. B. Conditional specification. These questions test the understanding of specification as a process in developmental biology, focusing on the role of intrinsic and extrinsic factors in determining cell fate. These concepts are critical for mastering developmental biology and preparation for exams like CSIR NET JRF Life Sciences. You said. Induction. ChatGPT said. Induction in developmental biology. Induction is a fundamental process in developmental biology in which one group of cells, the inducing tissue, influences the fate of another group of cells, the responding tissue, through signaling mechanisms. This interaction leads to a change in the developmental trajectory of the responding cells, causing them to adopt a different fate or undergo differentiation into a specific cell type. Induction is essential for coordinating the development of complex multicellular organisms, ensuring that cells and tissues develop in a coordinated manner to form functional organs and structures. Mechanisms of induction. Inductive signaling occurs through various molecular pathways, which typically involve the secretion of signaling molecules, often proteins, by the inducing tissue. These molecules bind to receptors on the surface of the responding cells, triggering intracellular signaling cascades that lead to changes in gene expression. This, in turn, can alter the fate of the responding cells. The most well-known signaling pathways involved in induction include the Notch, WNT, HETOG, TGF-beta, and BMP bone morphogenic protein pathways, among others. Morphogen gradients. In many cases, induction relies on the establishment of morphogen gradients, where signaling molecules are distributed in a concentration-dependent manner. For example, sonic hedgehog, SHISH, is a key morphogen that plays a crucial role in the patterning of the neural tube and limb development in vertebrates. The gradient of SHISH determines the fate of cells depending on the concentration of the morphogen they are exposed to, leading to the differentiation of various cell types. Juxtacrine signaling. In this form of induction, signaling occurs through direct cell-cell contact. This is seen in the Notch signaling pathway, where the Notch receptor on one cell interacts with ligands on an adjacent cell. This interaction leads to the activation of Notch within the responding cell, influencing its developmental fate. Paracrine signaling. Paracrine induction involves the release of signaling molecules by one group of cells into the surrounding extracellular space, which then affects nearby target cells. This type of signaling is particularly important during embryonic development, such as in the development of the heart and limb buds. The molecules involved in paracrine signaling can include growth factors, cytokines, and morphogens. Endocrine signaling. Although less common in developmental induction, endocrine signaling involves the release of signaling molecules, like hormones, into the bloodstream, allowing them to act on distant cells. This is particularly important in processes like sexual differentiation and metabolism, though it plays a smaller role in embryonic induction compared to other forms of signaling. Examples of induction in development. Neural induction. One of the best studied examples of induction is neural induction, where signals from the notochord, a rod-like structure in the early embryo, induce the overlying ectoderm to differentiate into neural tissue. This process is crucial for the formation of the neural plate, which eventually gives rise to the neural tube and the central nervous system. The signaling molecule sonic hedgehog, SHISH, secreted by the notochord, plays a central role in this process, initiating the differentiation of ectodermal cells into neural progenitors. Limb development. In vertebrates, the development of limbs is another classic example of induction. The apical ectodermal ridge, located at the tip of the limb bud, induces the underlying mesoderm to proliferate and elongate, leading to the formation of the limb. Signaling molecules such as fibroblast growth factors, FGFs, secreted by the play a key role in this induction process. Additionally, the zone of polarizing activity, ZPA, in the posterior part of the limb bud secretes SHISH, which helps to establish the anterior-posterior axis of the limb and determines the patterning of digits. Somite formation. In vertebrate embryos, the formation of somites, precursors to muscles, vertebrae, and dermis, is induced by signaling from the neural tube and the notochord. The induction of somite formation involves a complex interaction between signals from the presomitic mesoderm and the adjacent neural tissue, including molecules such as FGFs, WNTs, and Notch ligands. Role of induction in cell fate determination. Induction plays a crucial role in determining the fate of cells during embryonic development. It ensures that cells are directed to specific developmental paths in response to localized signaling cues. Without proper induction, developmental processes can be disrupted, leading to malformations or diseases. For example, in the development of the neural crest, cells at the edge of the neural tube are induced to migrate and differentiate into various cell types, including neurons, glial cells, and melanocytes. If induction signals are disrupted, it can lead to neural crest defects, which are associated with a range of developmental disorders. Induction in stem cells. In the context of stem cells, induction is used to direct pluripotent stem cells toward a particular lineage or tissue type. For instance, stem cells can be exposed to specific inductive signals that promote their differentiation into neurons, muscle cells, or cardiomyocytes, which is important for regenerative medicine. This process of directing stem cell differentiation is called directed differentiation and relies heavily on inductive signaling pathways. Conclusion. Induction is a cornerstone of developmental biology, orchestrating the proper formation and differentiation of tissues and organs. The process relies on a complex network of signaling molecules and pathways that provide spatial and temporal cues for cells to adopt specific fates. The ability of cells to respond to inductive signals is crucial for the proper development of an organism, and misregulation of these pathways can lead to developmental defects and diseases. Understanding induction is essential for advancements in stem cell therapy and regenerative medicine, where controlled cell fate decisions are central to therapeutic applications. Previous year questions. CSIR NET JRF 2016. Which of the following is a major signaling molecule involved in neural induction? A. Sonic hedgehog (CHISH). B. Fibroblast growth factor (FGF). C. Bone morphogenic protein (BMP). D. Epidermal growth factor (EGF). Answer. A. Sonic hedgehog (CHISH). CSIR NET JRF 2017. In vertebrate limb development, the apical ectodermal ridge induces the underlying mesoderm to form limb structures. Which of the following signaling molecules plays a key role in this process? A. WNT. B. Notch. C. Fibroblast growth factors (FGFs). D. Sonic hedgehog (CHISH). Answer. C. Fibroblast growth factors (FGFs). These questions reflect the importance of induction in developmental processes, especially regarding signaling molecules and their roles in various developmental stages. Understanding these signaling pathways is key for CSIR NET JRF preparation in life sciences. You said. Competence. ChatGPT said. Competence in developmental biology. Competence in developmental biology refers to the ability of a cell or group of cells to respond to an inductive signal. In other words, a competent cell is one that has the necessary molecular machinery, such as receptors, intracellular signaling pathways, and regulatory proteins, to respond to specific signaling molecules that influence its fate. Competence is a crucial concept in understanding how cells acquire specific fates during development, as it ensures that cells are capable of interpreting and responding to external signals that guide them toward particular developmental outcomes. Mechanisms of competence. Receptor availability. One of the key factors that determine a cell's competence is the presence of specific receptors on its surface. These receptors bind to signaling molecules, such as growth factors, morphogens, or hormones, which activate intracellular signaling cascades that ultimately influence gene expression. For example, in the process of neural induction, ectodermal cells become competent to respond to signaling molecules like sonic hedgehog (CHISH) or bone morphogenic proteins (BMPs), which direct them to differentiate into neural tissue. Without the appropriate receptors, a cell cannot respond to an inductive signal, and its fate cannot be properly determined. Intracellular signaling pathways. In addition to receptors, competent cells must possess the necessary intracellular machinery to translate the signals they receive into developmental changes. This involves the activation of specific signaling pathways, such as the Notch, WNT, hedgehog, TGF-beta, and MAPK pathways. The activation of these pathways leads to changes in gene expression that dictate the fate of the responding cells. For example, WNT signaling is involved in specifying anterior-posterior axis patterning during embryonic development, and the competence of cells to respond to WNT signals is crucial for proper body plan development. Epigenetic modifications. Epigenetic factors also play a role in a cell's competence. The chromatin structure of a cell influences its ability to respond to certain signals. For instance, cells that are in a more open chromatin state, with fewer repressive histone modifications, are more likely to be competent to respond to inductive signals. Conversely, cells with highly condensed chromatin might be less competent to activate certain genes in response to signals. This aspect of competence is particularly important in stem cell biology, where stem cells must be in a primed state to differentiate into specific cell types. Temporal competence. Competence is not a permanent state, it is often time dependent. During development, cells may become competent at specific stages of development, and their competence may change over time as they progress through different stages. For example, early embryonic cells are often competent to respond to a broad range of signals, while later in development, cells may become restricted in the types of signals they can respond to. This temporal regulation ensures that cells differentiate in a precise and coordinated manner, with developmental events occurring in the correct sequence. Localized competence. In many cases, competence is restricted to certain regions of an embryo or tissue. For example, in Drosophila melanogaster, the patterning of the embryo involves localized competence of cells along the Competence in developmental biology. Competence in developmental biology refers to the ability of a cell or group of cells to respond to an inductive signal. In other words, a competent cell is one that has the necessary molecular machinery, such as receptors, intracellular signaling pathways, and regulatory proteins, to respond to specific signaling molecules that influence its fate. Competence is a crucial concept in understanding how cells acquire specific fates during development, as it ensures that cells are capable of interpreting and responding to external signals that guide them toward particular developmental outcomes. Mechanisms of competence. Receptor availability. One of the key factors that determine a cell's competence is the presence of specific receptors on its surface. These receptors bind to signaling molecules, such as growth factors, morphogens, or hormones, which activate intracellular signaling cascades that ultimately influence gene expression. For example, in the process of neural induction, ectodermal cells become competent to respond to signaling molecules like sonic hedgehog (CHISH) or bone morphogenic proteins (BMPs), which direct them to differentiate into neural tissue. Without the appropriate receptors, a cell cannot respond to an inductive signal, and its fate cannot be properly determined. Intracellular signaling pathways. In addition to receptors, competent cells must possess the necessary intracellular machinery to translate the signals they receive into developmental changes. This involves the activation of specific signaling pathways, such as the Notch, WNT, hedgehog, TGF-beta, and MAPK pathways. The activation of these pathways leads to changes in gene expression that dictate the fate of the responding cells. For example, WNT signaling is involved in specifying anterior-posterior axis patterning during embryonic development, and the competence of cells to respond to WNT signals is crucial for proper body plan development. Epigenetic modifications. Epigenetic factors also play a role in a cell's competence. The chromatin structure of a cell influences its ability to respond to certain signals. For instance, cells that are in a more open chromatin state, with fewer repressive histone modifications, are more likely to be competent to respond to inductive signals. Conversely, cells with highly condensed chromatin might be less competent to activate certain genes in response to signals. This aspect of competence is particularly important in stem cell biology, where stem cells must be in a primed state to differentiate into specific cell types. Temporal competence. Competence is not a permanent state, it is often time dependent. During development, cells may become competent at specific stages of development, and their competence may change over time as they progress through different stages. For example, early embryonic cells are often competent to respond to a broad range of signals, while later in development, cells may become restricted in the types of signals they can respond to. This temporal regulation ensures that cells differentiate in a precise and coordinated manner, with developmental events occurring in the correct sequence. Localized competence. In many cases, competence is restricted to certain regions of an embryo or tissue. For example, in Drosophila melanogaster, the patterning of the embryo involves localized competence of cells along the anterior-posterior axis. This region-specific competence allows different parts of the embryo to respond to different signaling molecules, contributing to the formation of distinct structures like the head, thorax, and abdomen. Similarly, in limb development, cells in the zone of polarizing activity (ZPA) in the posterior part of the limb become competent to respond to sonic hedgehog signals, which help establish the patterning of digits. Competence and induction. Competence and induction are closely related concepts in developmental biology. Induction refers to the process where one group of cells, the inducing tissue, sends signals to another group of cells, the responding tissue, and competence refers to the ability of the responding cells to receive and interpret these signals. A cell can only undergo induction if it is competent to respond to the signaling molecules produced by the inducing tissue. For example, during neural induction, ectodermal cells must first become competent to respond to signals from the notochord, such as sonic hedgehog or noggin, before they can form neural tissue. Competence in stem cell biology. In stem cell research, competence is a critical factor in the controlled differentiation of stem cells into specific cell types. Pluripotent stem cells, such as embryonic stem cells (ESCs), are initially capable of becoming many different cell types. However, they must be primed to become competent to respond to specific signals that direct their differentiation. This can be achieved through the manipulation of signaling pathways, the application of growth factors, and changes in the culture environment. For example, in the differentiation of stem cells into neurons, it is essential to expose the cells to specific signaling molecules, such as FGFs and Notch ligands, that make the stem cells competent to adopt a neuronal fate. Competence in regenerative medicine. The concept of competence is also critical in regenerative medicine. In tissue repair and regeneration, it is essential that cells in the damaged area become competent to respond to the appropriate signaling cues. This is especially relevant in situations where stem cells are used to replace damaged or lost tissues. Ensuring that stem cells are competent to differentiate into the desired cell types is crucial for the success of regenerative therapies. Additionally, understanding how to induce competence in cells during tissue regeneration can provide insights into developing treatments for various diseases. Additionally, understanding how to induce competence in cells during tissue regeneration can provide insights into developing treatments for various diseases, such as neurodegenerative disorders, heart disease, and diabetes. Conclusion. Competence is a key concept in developmental biology, referring to the ability of cells to respond to inductive signals. It involves the presence of appropriate receptors, intracellular signaling machinery, and epigenetic factors that enable cells to interpret and act upon signals from their environment. Competence is essential for coordinated development, ensuring that cells differentiate into the correct cell types at the appropriate time and location. In stem cell biology and regenerative medicine, understanding and manipulating competence is crucial for directing the differentiation of stem cells into specific tissues for therapeutic purposes. Previous year questions. CSIR NET JRF 2018. Which of the following is required for a cell to respond to an inductive signal? Of the presence of specific receptors on the cell surface B, the presence of transcription factors alone C, the absence of signaling molecules D, the absence of receptor availability answer. Of the presence of specific receptors on the cell surface. CSIR NET JRF 2017. Competence of a cell to respond to a signaling molecule depends on which of the following? Of the presence of the appropriate receptors and signaling machinery B, the absence of epigenetic factor C, the lack of extracellular signaling molecules D, the inherent inability of the cell to differentiate answer. Of the presence of the appropriate receptors and signaling machinery. These questions test the understanding of the concept of competence in developmental biology, focusing on the cellular mechanisms that enable cells to respond to signaling cues. You said, determination. ChatGPT said. Determination in developmental biology. Determination is a crucial process in developmental biology that refers to the commitment of a cell or group of cells to a specific developmental fate, regardless of future environmental conditions. Once cells become determined, they are irreversibly set on a path towards a specific type of differentiation, meaning that they will eventually adopt a particular cell fate even if they are later transplanted into a different environment. This process is essential for the establishment of the various tissues and organs in a developing organism. Determination essentially locks in a cell's fate, preparing it for future differentiation and specialization. Key concepts of determination. Cell fate specification. Before determination, cells are specified, which means they have a predisposition to develop into a particular type of cell or tissue but are still flexible and capable of being influenced by external factors. During determination, this fate becomes fixed and irreversible, meaning the cell's developmental trajectory is no longer influenced by external signals. This specification and subsequent determination. During determination, this fate becomes fixed and irreversible, meaning the cell's developmental trajectory is no longer influenced by external signals. This specification and subsequent determination form the foundation for the orderly and structured development of multicellular organisms. Differentiation versus determination. It is important to distinguish between differentiation and determination. Differentiation is the process by which determined cells acquire the specialized structures and functions of a particular cell type. Determination, on the other hand, is a process that ensures a cell will eventually undergo differentiation into a specific cell type, but it does not involve the actual execution of the cell's specialized function. Determined cells will continue to undergo differentiation at the appropriate time, given the correct environmental cues. Molecular mechanisms of determination. Determination is governed by intricate molecular mechanisms, primarily involving the regulation of gene expression. Key transcription factors and signaling pathways play a pivotal role in determining the fate of cells. For example, during early embryogenesis in many organisms, morphogens signaling molecules like WNT, sonic hedgehog, and BMPs activate specific transcription factors that regulate the expression of genes that are crucial for the development of particular tissues. These transcription factors act as master regulators that orchestrate the developmental program of a cell. Intrinsic versus extrinsic factors. Determination is influenced by both intrinsic factors, like the genetic makeup of a cell and its history of gene expression, and extrinsic factors such as signaling molecules or the physical environment of the developing tissue. Intrinsic factors, including maternal mRNA or cytoplasmic determinants, play a significant role in early development. For example, during Drosophila oogenesis, maternal determinants like glycoid protein in the egg cytoplasm help establish the anterior-posterior axis and influence the fate of cells in the embryo. Extrinsic factors, like signals from neighboring cells, can either reinforce or modify the initial commitment made by a cell. Lineage-dependent versus induction-dependent determination. There are two primary mechanisms by which determination can occur: lineage-dependent and induction-dependent determination. Lineage-dependent determination occurs when cells inherit specific molecular signals from their ancestors. This means that a cell's fate is largely pre-programmed based on its lineage or ancestry. In contrast, induction-dependent determination relies on external signals from neighboring cells that direct the fate of a cell. For instance, during the formation of the neural tube, the notochord induces the overlying ectoderm to become determined to form neural tissue through the action of signaling molecules like sonic hedgehog. Examples of determination in model organisms. A classical example of determination is seen in the development of the C. elegans embryo. The M-SPE cell, in particular, is determined early in development to give rise to specific tissues, such as mesodermal and endodermal cells. In Drosophila, the specification and determination of the anterior-posterior axis and the development of segmental structures are regulated by the maternal gradients of bicoid and nonose proteins, which act as morphogens to establish the primary body plan. Changes in determination and reprogramming. While determination typically leads to irreversible commitment to a specific fate, recent advances in stem cell biology have shown that determination is not always a permanent state. Under certain conditions, it is possible to reprogram determined cells into different cell types, as seen in the creation of induced pluripotent stem cells (iPSCs). This reprogramming is driven by the introduction of key transcription factors such as OCTOBER4, SOX2, KLF4, and CMYC, which can reset the developmental clock of a determined cell and restore pluripotency, allowing the cell to differentiate into various cell types. This ability to reverse determination has significant implications for regenerative medicine and therapeutic interventions. Mechanisms and signaling pathways involved in determination. Various signaling pathways are integral to the process of determination. These include Notch signaling pathway. The Notch pathway is involved in lateral inhibition, which helps to establish specific cell fates within a population of cells. In developing tissues, Notch signaling ensures that cells receive specific signals from their neighbors that lock them into a particular developmental pathway. WNT beta-catenin signaling. This pathway plays a crucial role in determining the fate of mesodermal and ectodermal cells during early development. Activation of the WNT signaling pathway leads to stabilization of beta-catenin, which translocates to the nucleus and activates the transcription of target genes that influence cell fate decisions. BMP signaling. Bone morphogenic proteins BMPs are key regulators of cell fate determination, particularly in the formation of bone, cartilage, and other mesodermal derivatives. SMOD proteins act as intracellular mediators of BMP signaling, influencing gene expression patterns that guide cell differentiation. Hedgehog signaling. The hedgehog pathway, particularly sonic hedgehog (CHH), is essential in the determination of cells in the neural tube and limb development. It regulates the expression of specific transcription factors that determine the fate of surrounding tissues. Conclusion. Determination is a fundamental process in developmental biology that ensures the establishment of cell fates within a developing organism. It is influenced by complex networks of genetic and signaling factors that ensure the correct tissues and organs are formed at the appropriate time. This process is essential for proper organismal development and is regulated by both intrinsic genetic factors and extrinsic signaling cues. As research in developmental biology progresses, our understanding of determination will continue to advance, with significant implications for stem cell biology and regenerative medicine. Previous year questions. CSIR NET JRF 2019. Which of the following is directly involved in the determination of the ectodermal cells to form neural tissue? A. FGF. B. BMP. C. Sonic hedgehog. D. Notch. Answer. C. Sonic hedgehog. CSIR NET JRF 2018. The process by which a cell is irreversibly committed to a particular developmental phase. Which of the following is directly involved in the determination of the ectodermal cells to form neural tissue? A. FGF. B. BMP. C. Sonic hedgehog. D. Notch. Answer. C. Sonic hedgehog. CSIR NET JRF 2018. The process by which a cell is irreversibly committed to a particular developmental phase. The process by which a cell is irreversibly committed to a particular developmental phase is known as a differentiation. B. Induction. C. Specification. D. Determination. Answer. D. Determination. These questions test the understanding of determination, its regulatory mechanisms, and its importance in developmental biology. Understanding of determination, its regulatory mechanisms. Understanding of determination, its regulatory mechanisms, and its importance in developmental biology. To understanding of determination, its regulatory mechanisms, and its importance in developmental biology. You said. Differentiation. ChatGPT said. Differentiation in developmental biology. Differentiation is the process by which unspecialized cells, typically stem cells or progenitor cells, acquire specialized structures and functions, becoming distinct cell types. This is a fundamental aspect of development, as it ensures that the various cells in a multicellular organism perform specific roles. Differentiation is a key step in the formation of tissues, organs, and the overall body plan. It involves both changes in gene expression and cellular morphology, allowing a single fertilized egg to give rise to the diverse range of cell types that make up the organism. Key aspects of differentiation. Gene expression changes. Differentiation is driven by changes in gene expression. A specific set of genes is activated or repressed in different cells, resulting in the production of proteins that define the structure and function of each cell type. For example, a mesodermal cell might differentiate into a muscle cell, which involves the activation of muscle-specific genes like myosin and actin, proteins that are responsible for contraction. These changes are regulated by transcription factors, signaling molecules, and other epigenetic factors that orchestrate the cellular machinery. Stages of differentiation. Differentiation occurs through several stages, starting from a pluripotent or multipotent stem cell, which has the potential to differentiate into multiple cell types. The early stages of differentiation are characterized by a broad potential to form different tissues, while later stages involve a more restricted fate, where the cell becomes more specialized. For instance, during hematopoiesis, hematopoietic stem cells differentiate into various blood cell types, including red blood cells, white blood cells, and platelets, each of which has specific functions in the body. Intrinsic and extrinsic factors. Differentiation is influenced by both intrinsic factors, internal cell programming, and extrinsic factors, external signals. Intrinsic factors include cytoplasmic determinants inherited from the parent cell, while extrinsic factors are signaling molecules, such as growth factors, hormones, and morphogens, which interact with receptors on the surface of the cell. These external signals can induce the expression of certain genes, leading to differentiation. For example, the signaling molecule Notch plays a critical role in determining whether a progenitor cell becomes a neuron or a glial cell. Morphological changes. Along with gene expression changes, differentiation involves significant changes in cell morphology. For example, muscle cells elongate to form fibers, while epithelial cells form sheets. These structural changes are essential for the specialized functions of the differentiated cell. In neurons, differentiation involves the formation of dendrites and axons, enabling communication across the nervous system. Commitment to cell fate. Differentiation is the culmination of processes such as determination and specification. Once a cell has been determined, it will begin to differentiate according to its predetermined fate. The commitment of a cell to differentiate is usually irreversible, meaning once a cell has undergone differentiation, it cannot change into a different cell type. For example, once a mesodermal cell differentiates into a muscle cell, it will not change back into a skin cell. Stem cells in differentiation. The role of stem cells in differentiation is paramount. Stem cells are undifferentiated cells that have the ability to differentiate into various specialized cell types. They are classified into totipotent, pluripotent, multipotent, and unipotent based on their potential to differentiate into different cell types. Embryonic stem cells (ESCs) are pluripotent, meaning they can differentiate into almost any cell type in the body, while adult stem cells are usually multipotent, restricted to specific tissues, e.g., hematopoietic stem cells differentiate into blood cells. Induced pluripotent stem cells (iPSCs) are somatic cells that have been reprogrammed to an embryonic-like pluripotent state and can differentiate into various cell types, offering significant potential in regenerative medicine. Notch and WNT signaling in differentiation. Two of the most important signaling pathways in differentiation are the Notch and WNT pathways. Notch signaling is involved in cell fate determination, especially in the nervous system, where it helps regulate whether cells become neurons or glial cells. WNT signaling, on the other hand, is involved in the regulation of cell proliferation, migration, and differentiation during various developmental processes, including the formation of the nervous system, limbs, and other tissues. These pathways highlight how external signals are crucial for guiding the differentiation of cells. Epigenetic regulation of differentiation. Differentiation is regulated by epigenetic modifications, such as DNA methylation and histone modification, which control the accessibility of DNA and the activation of specific genes. These modifications are reversible and can be influenced by the environment, signaling molecules, and developmental cues. The chromatin remodeling process ensures that the necessary genes are activated and others are silenced to allow for the correct differentiation of cells. Types of differentiation. Terminal differentiation. In terminal differentiation, a cell reaches its final, specialized form and function. These cells typically do not divide further and perform specific functions in the body. For example, erythrocytes, red blood cells, undergo terminal differentiation, losing their nuclei and becoming specialized for oxygen transport. Plasticity and transdifferentiation. While differentiation is often thought of as a one-way process, types of differentiation. Terminal differentiation. In terminal differentiation, a cell reaches its final, specialized form and function. These cells typically do not divide further and perform specific functions in the body. For example, erythrocytes, red blood cells, undergo terminal differentiation, losing their nuclei and becoming specialized for oxygen transport. Plasticity and transdifferentiation. While differentiation is often thought of as a one-way process, certain cells exhibit plasticity, meaning they can undergo reprogramming to become different types of cells. This is known as transdifferentiation. For example, under certain conditions, liver cells can transdifferentiate into pancreas cells. Lineage-restricted differentiation. Some differentiated cells retain the ability to differentiate further within a particular lineage. Neural stem cells, for example, differentiate into various types of neurons and glial cells, but they are restricted to the neural lineage. Clinical relevance. The process of differentiation has significant implications for regenerative medicine and cancer therapy. Stem cell-based therapies are designed to exploit the potential of undifferentiated cells to replace damaged or diseased tissues. However, the controlled differentiation of stem cells is crucial to avoid the development of tumors. On the other hand, in cancer, differentiation is often disrupted, with cancer cells failing to fully differentiate, leading to uncontrolled cell division and tumor formation. Conclusion. Differentiation is a highly regulated, complex process that is fundamental to the development of multicellular organisms. It ensures that cells become specialized to perform the specific functions required for the proper functioning of tissues and organs. The study of differentiation is essential for understanding development, disease, and the potential for regenerative medicine. Previous year questions. CSIR NET JRF 2018. Which of the following processes ensures that a cell becomes specialized to perform a specific function? A. Determination. B. Differentiation. C. Induction. D. Commitment. Answer. B. Differentiation. CSIR NET JRF 2017. What is the role of transcription factors in cell differentiation? A. To maintain the pluripotency of the cell. B. To activate specific gene expression patterns for differentiation. C. To inhibit the cell cycle. D. To support cell division. Answer. B. To activate specific gene expression patterns for differentiation. You said. Morphogenic gradients. ChatGPT said. Morphogenic gradients in developmental biology. Morphogenic gradients are a key concept in developmental biology, essential for patterning and organizing the spatial arrangement of tissues and organs during development. They refer to the distribution of signaling molecules, known as morphogens, that vary in concentration across a developing tissue or organism, leading to different cellular responses at different concentrations. These gradients are crucial for guiding the differentiation of cells, determining their fate, and orchestrating the proper development of structures in multicellular organisms. Morphogenic gradients help establish the anterior, posterior, dorsal ventral, and left-right axes in organisms, contributing to the precise formation of tissues and organs. Basic concept and mechanism. Morphogens are signaling molecules that exert their effects in a concentration-dependent manner. The gradient of these molecules forms as they are secreted by a localized source, such as a group of cells, and diffuse through the surrounding tissue. The concentration of the morphogen decreases as the distance from its source increases, creating a gradient. Cells within this gradient respond differently depending on the concentration of the morphogen they encounter. These responses can include changes in gene expression, cellular differentiation, or movement, which collectively contribute to the formation of specific tissues or structures. A classical example of a morphogen gradient is a wingless WNT gradient in the developing Drosophila embryo, which helps establish the anterior-posterior axis. Another well-known example is the sonic hedgehog gradient, which is crucial for patterning the neural tube and limbs invertebrates. Key features of morphogenic gradients. Concentration-dependent effects. Morphogens exert their effects in a concentration-dependent manner, meaning that different concentrations of the morphogen lead to different cellular responses. For example, high concentrations of a morphogen might induce one type of cell fate, while lower concentrations might induce a different fate. This principle is exemplified by the gradient of sonic hedgehog where different concentrations of in the neural tube direct the formation of distinct neuronal subtypes. Thresholds of response. Cells within a morphogen gradient can exhibit different responses depending on the concentration of the morphogen they encounter. At higher concentrations, cells may undergo a specific developmental program, while at lower concentrations, they might follow a different program. These thresholds help define cell fate decisions in the developing embryo. For example, in limb development, the gradient determines the identity of cells in the posterior part of the limb, with high levels specifying digits and low levels determining the formation of the anterior structures. Source and diffusion. The source of morphogen production can be a localized group of cells or an external signal, and the gradient forms as the morphogen diffuses across the surrounding tissue. The rate of diffusion and the degradation of the morphogen help shape the gradient. In addition to diffusion, the modulation of morphogen transport, as well as its binding to extracellular matrix components or receptors, can regulate the extent and shape of the gradient. Feedback mechanisms. Morphogen gradients are often regulated by feedback loops, both positive and negative. For instance, in the case of Drosophila wing development, the morphogen decapitoplegic DPP forms a gradient, and the feedback between the morphogen and its receptors helps refine the gradient, ensuring that cells respond appropriately to the morphogen at the correct stage of development. This feedback loop helps maintain the integrity and stability of the gradient during the progression of development. Gradient interpretation. Cells interpret the gradient based on the concentration of morphogens they are exposed to, activating specific transcription factors and signaling pathways. These transcription factors then activate or repress genes involved in differentiation and development. For instance, in the Drosophila embryo, bicoid forms a gradient along the anterior-posterior axis, and cells interpret this gradient to establish the head and tail structures. Regulation of morphogen gradients. The formation and maintenance of morphogenic gradients are tightly regulated through several mechanisms, including the localization of morphogen sources, diffusion rates, and morphogen degradation. Some gradients, such as the BMP bone morphogenic protein gradient, are controlled by antagonists that bind to the morphogen, limiting its diffusion and ensuring its proper distribution. Examples of morphogenic gradients. Sonic hedgehog gradient. One of the most well-studied examples of a morphogen gradient is sonic hedgehog In vertebrates, is crucial for the development of the neural tube, limb buds, and various other tissues. In the neural tube, a gradient of establishes the dorsal ventral axis, with high concentrations of specifying the ventral neurons and lower concentrations specifying dorsal neurons. Similarly, in limb development, the gradient defines the posterior region of the limb bud, influencing digit formation. Bicoid gradient in Drosophila. In Drosophila embryos, the bicoid protein forms a gradient from the anterior end to the posterior end. This gradient is critical for determining the anterior structures of the embryo, including the head and thorax. Cells at the anterior end of the embryo, exposed to higher concentrations of bicoid, differentiate into head structures, while cells at the posterior end, exposed to lower concentrations, form tail structures. Wingless WNT gradient. In Drosophila and other organisms, the WNT signaling pathway, through the morphogen wingless, plays a central role in establishing anterior-posterior polarity during embryonic development. The wingless gradient helps pattern tissues such as the wing imaginal discs in Drosophila and is crucial for limb development in vertebrates. Implications in development and disease. The proper formation of morphogenic gradients is essential for normal development, as these gradients control cell fate, pattern formation, and tissue organization. Disruptions in gradient formation can lead to developmental disorders and diseases. For example, mutations affecting signaling can result in conditions like holoprosencephaly, a congenital disorder where the forebrain fails to divide properly into two hemispheres. Furthermore, morphogenic gradients play a role in cancer development, as abnormal gradients can lead to incorrect cell patterning and unregulated cell growth. Tumors may arise when cells fail to respond correctly to morphogenic signals, resulting in uncontrolled differentiation and proliferation. Conclusion. Morphogenic gradients are fundamental to the development of multicellular organisms. They guide the differentiation of cells, pattern the body plan, and contribute to the formation of tissues and organs. The concentration-dependent nature of morphogen signaling, combined with feedback mechanisms and intricate regulation, ensures the precise and coordinated development of an organism. Understanding these gradients and their regulation provides critical insights into normal development as well as the pathogenesis of developmental diseases and cancers. Previous year questions. CSIR NET JRF 2016. Which of the following is a key feature of morphogen gradients during development? A. High concentrations of morphogens induce different cellular responses than low concentrations. B. Morphogens diffuse equally across tissues, leading to uniform cellular responses. C. Morphogen gradients are unaffected by feedback mechanisms. D. Morphogens are secreted by cells but do not influence nearby cells. Answer. A. High concentrations of morphogens induce different cellular responses than low concentrations. CSIR NET JRF 2019. In the context of developmental biology, what is the role of a morphogen gradient in tissue patterning? It provides a uniform concentration of signaling molecules. B. It determines the anterior-posterior axis by varying the concentration of signaling molecules across tissues. C. It promotes stem cell proliferation. D. It helps in the formation of blood vessels. Answer. B. It determines the anterior-posterior axis by varying the concentration of signaling molecules across tissues. You said. Self-ate. ChatGPT said. Self-ate in developmental biology. Self-ate refers to the specific developmental outcome or identity that a cell adopts during the process of embryogenesis, influenced by intrinsic factors, genetic information, and extrinsic factors, signals from the surrounding environment. Self-ate determination is a fundamental aspect of developmental biology and is essential for the proper formation of tissues, organs, and the overall body plan of multicellular organisms. The study of self-ate involves understanding how cells decide what function or identity they will take on, whether it be part of the nervous system, muscle tissue, skin, or other specialized cells. Mechanisms of self-ate determination. Self-ate is determined by a combination of molecular, biochemical, and mechanical signals that guide cells through different stages of differentiation. These mechanisms can be broadly categorized into intrinsic and extrinsic factors. Intrinsic factors. These are the internal components of the cell, such as the expression of specific genes, transcription factors, and RNA molecules that regulate the developmental path of the cell. These factors are influenced by the cell's position in the embryo or tissue and can often be inherited from parent cells during division. For example, the bicoid protein in Drosophila embryos is an intrinsic factor that helps determine the anterior-posterior axis of the embryo, setting the stage for the differentiation of cells into head or tail structures. Extrinsic factors. These are external signals from neighboring cells or the environment that influence a cell's fate. These signals are often transmitted through cell-cell communication and involve the action of morphogens.